Hello students, welcome back to the channel. In this video, we are going to study about genetic drift. Now see, genetic drift is what? Actually, it is a random change event. Because of some random events, there is a change in the allele frequency. And it is non-selective change. Okay, so because of some random chance event, allele frequency is changing and it is non-selective, and main thing is this key, it is equally affecting all members of the population. One allele may be eliminated, and elimination it might be by chance, whether it is beneficial or it is harmful or of no particular advantage. So you can say genetic drift, which is generally operable in the small population size, it can lower down genetic diversity and it makes survivability more difficult, though it creates genetic difference among different populations. Now, right now only we discuss that it is seen in the population of a small size. More effect will be significant in the small size population only. Now, two important causes of genetic drift is one is founder effect, which lead to the new population. Another is bottleneck effect. Now, let us understand one by one. Now, see founder effect. Let us assume a small group of existing population. That existing population, a small group that is known as founder, they leave the original large population to colonize new areas and they will breed with the member of this new population only. So as a result, the new population, which is colonizing unoccupied and isolated area, prevents the gene flow between the initial and new population. So slowly, they will make a new gene pool and we will say allele frequency have changed. So new population has evolved. Is it clear? Now you can take an example. Suppose from here, randomly you are taking it, chance isolation is going on and you have isolated only by chance red and yellow color and see new population is different from the original population. Clear beta? Now see next example. Again, if you see here, this is the ancestral population, okay, and group. This small group is changing and it changes and see the changes. See here. Now, if you see the population in the original population, what is the frequency of red allele? Red allele was 0.3. And what is the frequency of green allele? It was it was equal 0.5 and this is also 0.5. But now if you see here, after this changes, see after split, red was 0.2 and green was 0.8. Clear? Immediately after split, you are finding this. After first generation, if you see now what is going on, after first generation, you are finding, check, ki red is 0.4 and green is 0.6. And founder population is actually, if you see red is 1. So this is the way you can see founder effect means, once again, if you want to see back, a small group of existing population, small group, leaves the original large group to colonize new area. That is known as founder, and this effect because of which population become entirely different from the original population, we say that is known as founder effect. Clear beta? So this is founder and this is founder effect. Now bottleneck. Because of the fluctuations in the environment, like depletion in the food supply or outbreak of disease, a population may either rapidly or markedly decrease from the time to time. The population is said to go through the bottleneck, during which genetic drift can occur in a small population of survivor. Now, when the population increases again in the size, what will happen? We will find many allele frequency is now different from the original population. If you see here, this was the original population and survivor are allowed to come only this much. Suppose surviving is actually just like a bottleneck, you are finding it, effect is working. Only few individuals are able to survive. To check the surviving population will slowly, when they multiply, they will appear lot of difference from the original population. So as the population when again increase in size, many allele frequency may be quite different from the original population. And they have hypothesized that genetic variation in the cheetah was reduced by bottleneck effect when at the end of ice age some 10,000 years ago. Cheetah has nearly become extinct because of the overhunting by humans, and surviving cheetah which are left, they are having very reduced genetic variability. So let us just revise once again. See what is genetic drift. Now you can get the answer. Okay, what is genetic drift? You will give it. Genetic drift is because of the chance event, and chance event will is non-selective, okay, and it will change the allele frequency. Now, allele which is eliminated, it is occurring by chance. It is not beneficial is eliminated or harmful is eliminated. It is purely by chance. Main problem is it can lower down the genetic diversity and make survivability more difficult. Though, obviously, it will create genetic differences. Obviously, it will create genetic differences among different population, just like we have observed in the founder effect and we have studied in the bottleneck effect. So, genetic drift can be because of two reasons. Two important causes of genetic drift is one is founder effect, another is your bottleneck effect. So, I hope so this video might be helpful for you. Thanks for watching.